Dear students, welcome to the first session of Nucleic Acids of 6th Semester Chemistry DSc 2B, University of Mysore. Contents of this video are definition of nucleic acids, occurrence of RNA and DNA, components of nucleic acids, history of nucleic acids. One of the major scientific achievements of 20th century is the discovery of nucleic acids and their biological function. Nucleic acids were first isolated by Friedrich Meiser in 1853 from the nuclei of pus cells present in hospital bandages. However, he called these compounds nucleins. Altman named them in 1889 as nucleic acids because of their acidic nature. Definition of nucleic acids. Nucleic acids are important biological polymers present in living cells. They are responsible for the storage and transmission of genetic information. They have molecular weights in the range of 10 to the power 9. Classification of nucleic acids. There are two types of nucleic acids. One is deoxyribonucleic acid, DNA. Second one is ribonucleic acid, RNA. DNA encodes the hereditary details and controls the growth and division of the cells. The genetic information stored in DNA is then transcribed into RNA, and the details in RNA are then translated for the synthesis of the proteins. Occurrence of nucleic acids. RNA is found in the nucleus as well as cytoplasma of a cell. It is found in nucleolus, nucleoplasma, and chromatins present inside the nucleus. It is also found in ribosomes present in cytoplasma. DNA is found in the nucleus in the form of nucleoproteins. It is also present in mitochondria, chloroplasts, and the other self-replicating organelles. DNA is also present in some viruses. Components of nucleic acids. Nucleic acids are the polymers of nucleotides held by 3' and 5' phosphate bridges. Nucleotides are composed of a nitrogenous base, pentose sugar, and phosphate group. Functions of nucleotides are they are building blocks or monomeric units in the nucleic acid structure, structural components of some coenzymes of vitamin B, as energy currency of the cells, as second messenger, and the control of metabolic reactions. Structure of nucleotides. Nucleotides on hydrolysis gives a molecule of pentose sugar, nitrogen base, and a phosphate residue. Coming to the pentose sugar present in RNA is D-ribose and deoxyribose in DNA. Ribose and deoxyribose differ in the structure at second carbon atom. Deoxyribose has one oxygen less at C2 compared to ribose. These are the structures of ribose and deoxyribose. In ribose, we have OH group at second position. Here at the second position, we don't have oxygen. Second is nitrogen bases. The nitrogenous bases are derivatives of pyrimidine and purine. The numbering of the ring atoms of pyrimidines and purines is illustrated as follows. In pyrimidine, the numbering is in the clockwise direction. In purine, in this ring it is anti-clockwise, in this ring it is clockwise. The pyrimidine bases are cytosine, thymine in DNA, cytosine, uracil in RNA. The two major purine bases are adenine and guanine, present in both DNA and RNA. This is the summary. In DNA, we have adenine and guanine. Pyrimidine bases are cytosine and thymine. In RNA also, purine bases are same. Pyrimidine bases are cytosine and uracil. Structures of pyrimidine and purine bases. So these are the structures of purine bases: adenine, guanine, pyrimidine, thymine, cytosine and uracil. Third one is phosphate residue. The phosphate residues in nucleic acids are derived from phosphoric acid. Nucleoside, a compound with a nitrogen base bonded to ribose or deoxyribose, is called nucleoside. Nucleoside is nothing but nitrogen base and sugar. Nucleotide and nucleic acid nomenclature. So base, these are the bases, nucleosides, nucleotides, and this is the nucleic acid. So purine bases are adenine and guanine. Corresponding nucleosides are adenosine. Nucleotide is adenylate in RNA. So nucleoside deoxyadenosine. Nucleotide is deoxyadenylate, present in DNA. If the base is guanine, nucleoside is guanosine. Guanylate is nucleotide, present in RNA. Deoxyguanosine is nucleoside and deoxyguanylate is nucleotide, present in DNA. Pyrimidine bases are cytosine, thymine and uracil. So corresponding nucleoside is cytidine, nucleotide is cytidylate, present in RNA. So nucleoside deoxycytidine and nucleotide is deoxycytidylate, present in DNA. If it is thymine, nucleoside is thymidine or deoxythymidine. Nucleotide is thymidylate or deoxythymidylate, present in RNA. Uridine is the nucleoside of uracil. Uridylate is nucleotide, present in RNA. MCQs. First one is, if the compound does not contain the phosphate group, it is known as nucleoside. Second is, nucleotides are linked together to form nucleic acid through which type of bond? Answer is phosphodiester bond. How many nucleotide bases occur in nucleic acid? That is 5. Where is RNA present? In both nucleolus and cytoplasma. Which of the following is not present in RNA? That is thymine. Which of the following is pyrimidine nucleotide? All of the above. Thank you.